Imagine being born with a deadly genetic disorder, one that's so rare there is no drug, no cure, and no standard treatment. Now imagine doctors editing your DNA sequence to save your life. That's exactly what happened to KJ Muldoon, a baby boy treated with the first personalized gene editing drug ever created. This isn't science fiction, it's happening now, and this marks the beginning of a new era in medicine, where your genes aren't your fate, but something we can rewrite. Gene editing is a revolutionary field in molecular biology that allows scientists to make precise changes to an organism's DNA. These changes can correct harmful mutations or introduce new traits directly into the genetic level. Over the past decade, gene editing has rapidly advanced with the advancement of powerful tools. The most well-known is CRISPR-Cas9, but it's not the only one. Other methods include base editors, prime editors, and older tools like zinc finger nucleases and TALENs. Still, CRISPR-9 has become the face of gene editing for a reason. It's simple, it's precise, and forms the foundation for many of today's breakthroughs, including one that is used to treat KJ Muldoon. CRISPR-Cas9 is one of the most groundbreaking gene editing tools. It allows scientists to make precise cuts in DNA in exact locations. The system relies on an enzyme called Cas9, essentially a molecular scissors basically, which is guided by a custom piece of RNA that matches a specific DNA sequence. Compared to old gene editing tools, it's faster, it's more efficient, and far more accessible, making it a game changer for both researchers and clinical medicine. Base editing is a newer, even more refined method of gene editing. Unlike CRISPR-Cas9, which cuts both strands of the DNA and relies on the body's own mechanism to repair that break, base editing skips that cutting entirely. Instead, it changes a single DNA base pair, like switching a typo in the Word document, without breaking the DNA strand at all. This results in fewer errors and greater accuracy. It makes it ideal for correcting diseases caused by single-point mutation, like sickle cell anemia and cystic fibrosis. Gene editing is no longer just experimental. The FDA has already approved groundbreaking therapies like Casgevy, a CRISPR-based therapy that's used for sickle cell disease and beta thalassemia. Likewise, Skysona, a lentiviral gene therapy that's used for cerebral adrenoleukodystrophy. These treatments don't just manage symptoms, they aim to correct the genetic root cause. And in KJ Muldoon's case, well, it goes one step further. His therapy is the first example of a gene editing drug specifically designed for one patient. Now that we have somewhat of an understanding of what gene editing is and how it works at a very high level, let's dive deeper into the specific methods that's used in the clinical setting and how they were applied in this groundbreaking case of KJ Muldoon. Before we dive deep into CRISPR-Cas9 and base editing and how everything works on a deeper level, we first need to understand the human genome, a quick overview. So the human body is made up of trillions of cells, each containing a nucleus. Inside the nucleus are chromosomes, long strands of DNA that carry our genetic information. While some sections of DNA code for proteins, much of it is actually non-coding and involves regulation or structural roles. So DNA is built from a sugar phosphate backbone and 4 nitrogenous bases: adenine, thymine, cytosine, and guanine. These base pairs A to T and C to G form the rungs of the DNA ladder. Genes are specific sequences of these bases that provide instructions for building proteins, which carry out vital cellular functions. When mutations disrupt these sequences, diseases can result. Now that we understand how genetic information is stored and how mutations can lead to disease, let's take a look at one of the most powerful tools that we've developed to fix these mutations. In order to understand how this system works in humans, we first need to understand how this works in bacterium, which is where it was originally discovered. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It's not a machine, it's not a protein, it's actually just a section of DNA found in bacteria. Think of it like a genetic filing cabinet with empty slots called spacers that bacteria use to store mugshots of past viral invaders. When a virus attacks a bacterium, the bacterium can capture a snippet of the viral DNA and insert it into one of these CRISPR spacer slots in its own genome. This captured viral sequence acts as a molecular memory. Later, if the same virus or a closely related one attacks again, the bacterium uses this stored sequence to produce a guide RNA. This RNA is complementary to the viral DNA, meaning that it can base pair with it precisely. The guide RNA then binds to a protein called Cas9, a molecular scissors. Together they form a complex, a surveillance complex. When the guide RNA finds a matching sequence in the viral DNA, Cas9 cuts the viral genome at that exact position, rendering the virus inactive. In short, the bacteria recognizes the target and destroys the invader using a system that looks remarkably programmable. Scientists took this naturally occurring immune system and repurposed it for gene editing in humans. Here's how it works in the lab. Researchers design a guide RNA that's complementary to a specific sequence in the human genome, often a region that contains a disease-causing mutation. The synthetic guide RNA is then combined with Cas9 to form the complex. Together, the guide RNA directs the Cas9 to the exact location in the DNA where the edit needs to happen. Once they bind to the target site, Cas9 makes a double-stranded cut in the DNA. The cell responds by activating a natural DNA repair mechanism. During the repair window, scientists can do a few things. They can disrupt the gene, introducing small deletions. They can insert a new gene or correct a mutation by providing a healthy template sequence. So to summarize, CRISPR refers to the system, specifically the DNA sequence that produces the guide RNAs, and Cas9 is the enzyme that cuts the DNA. In simple terms, think of CRISPR as the GPS that finds a target, and think of Cas9 as the scalpel that does the cutting. On the other hand, base editing offers even finer control. Instead of cutting both DNA strands, it uses a modified Cas9 enzyme called nickase to make a single strand nick. Then a specialized enzyme chemically changes one DNA base pair to another, like swapping out a single letter in a sentence. This avoids the risk of DNA breaks and is ideal for correcting single point mutations. In KJ Muldoon's case, KJ Muldoon was born with a CPS1 deficiency, a rare genetic disorder caused by mutation in the CPS1 gene. The gene encodes for an enzyme in the liver that removes ammonia, a toxic byproduct of protein metabolism. Without this enzyme, ammonia builds up in the blood and it can lead to seizures, coma, and death. Researchers use base editing to precisely correct the single point mutation in KJ's liver cells. The therapy was delivered via lipid nanoparticles. These are tiny fat particles that were used as carriers or transporters to transport the editing machinery directly to his liver. Some of KJ's liver cells successfully incorporated this corrected gene and began producing functional CPS1 enzyme, which could break down the ammonia byproduct. Well, what's the result? KJ showed significant improvement, including better protein tolerance and development of gains. This was a major leap forward for a condition that was previously managed only through strict diet or a liver transplant. While not all liver cells were edited and did not incorporate the new genetic modification, additional treatments may be needed in his case as he grows. KJ's case marks a historic first, a personalized gene editing therapy tailored to a single patient. Gene editing tools like CRISPR-Cas9 and base editors are powerful, but as KJ's story shows, they also come with technical, practical, and ethical challenges that we still need to solve. Let's take a look at what's standing in the way of broader use. Gene editing holds transformative potential, but it also comes with serious technical and real-world limitations. There are two things that I will discuss: scientific and ethical challenges. Number one, scientific challenges. So scalability. So many current therapies, like the one in KG's, are built for a single point mutation. Personalizing treatment on a larger scale is much more complex, slow, and expensive. There are also regulatory hurdles, and individualizing manufacturing also is very slow to progress. Number two, cost of therapy. Treatments can cost hundreds of thousands of dollars, even up to millions of dollars per treatment. For example, Casgevy, the FDA-approved CRISPR therapy for sickle cell anemia, costs around $2 million. This raises the concern for affordability and access. Who can actually afford this type of treatment? Number three, delivery limitation. Delivering gene editing tools into the right cells, into the right tissue, remains a major hurdle. Current delivery platforms, like the lipid nanoparticles, may not reach all target cells. It can provoke an immune response, and it may be limited by redosing. So not all cells will incorporate the genetic alteration, which will lead to redosing of the medication. So gene editing doesn't just raise scientific questions, it forces us to consider what kind of future we want to create. So treating a disease, number one. So editing somatic cells to treat or cure a genetic disease like sickle cell anemia or CPS1 deficiency is widely supported or ethically acceptable. These are therapeutic, non-inheritable changes intended to restore health. But preventing a disease is somewhere in the gray area. So editing embryos or editing germline cells to prevent disease, like BRCA mutations in breast cancer or Huntington's, raises ethical concerns. How does this affect the future generation? And the future generation is unable to give their consent. Lastly, human enhancement. This is definitely and broadly accepted as unethical. Editing genes for non-medical traits, things like intelligence, height, appearance, this crosses into eugenics. And there are some risks that come with this. You're widening the inequality gap. Editing traits we don't fully understand comes with effects that we don't understand, and it's a slippery slope of genetic optimization. So what is the bottom line? Treating a disease is widely accepted as ethical. Preventing the disease is debatable, somewhere in the gray area. And then enhancing traits is widely accepted as unethical. So while gene editing is one of the most powerful and medical technologies of our time, it must be approached with caution, humility, and oversight. In this final section, I will explore what this means for the future and why KJ Muldoon's story is just the beginning. I want to finish this video by mentioning that gene editing therapy is just in its infancy. Right now, we're very limited to treating rare single gene disorders like CPS1 deficiency, conditions that are relatively straightforward compared to complex polygenic diseases like Alzheimer's disease or diabetes. KJ Muldoon's story is a breakthrough, but not a cure. His case reminds us that technology is real, it's evolving, and profoundly human. But for the first time, we've rewritten DNA to treat a disease tailored to a single child's genome. The science may offer a tool, but the future, that's a story that we're still rewriting.